Hi, and welcome to Meet the Microbiologist, the podcast about the people who discover, innovate, and advance the field of microbiology. I'm your host, Ashley Hagen, and today I'm joined by Dr. Michael Lynch, Director of the Center for Mechanisms of Evolution at Arizona State University, and Dr. Vaughn Cooper, Professor of Microbiology and Molecular Genetics at the University of Pittsburgh. Welcome to the show, Mike and Vaughn.
Thanks very much.
Thank you.
So, the journey of life on Earth, from non-living chemical compounds to early unicellular life to the vast diversity of organisms that we see today, is nothing short of extraordinary. And since none of us were around to witness it, the origins of life have captured the minds of scientists for ages. So today we're going way back to examine what is known about the history of life on our planet, which, as it turns out, is reaching unprecedented levels thanks to advances in technology and cross-disciplinary collaboration. This episode is brought to you by the American Academy of Microbiology, a think tank at the American Society for Microbiology, and the Gordon and Betty Moore Foundation, which has been dedicated to advancing scientific discovery for the past 25 years. A recent project organized by the Academy and funded by the Moore Foundation brought together rock star scientists from an array of scientific disciplines to contribute knowledge to questions about early microbial life. And that project culminated in an over 200-page report, which I will link in the show notes. You can find on asm.org/MTM. Mike and Vaughn are the co-chairs of the steering committee for this project. So, Mike and Vaughn, before we get started and dive into the science, I want to set the stage for our listeners. And I'd love to talk about how the two of you met and where the idea for this project came from.
I don't really know how we met. Do you remember?
Yeah, it goes back to when you had a big project measuring mutation rates in bacteria.
Oh, that I remember.
So that's how we originally met. So that project just actually Mike mentioned in today's presentation, that big project tested this idea that mutation rates vary across life, right? And it raises questions about how mutation rates evolve and why they differ. It's one of these deep questions that kind of motivates us in the field and got us thinking deeply about how this all got going. So I had the good fortune to work with Mike on that project that he led for a number of years. When director of the Academy was approached by the Moore Foundation about the possibility of putting this project together under the auspices of the Academy, and then Wynn tapped me maybe as the evolutionary biologist that he knew close at hand. He said, "Should we do this?" And I said, "Absolutely, we should do this." But the problem was we needed to come up with a we. And my first inclination was I needed to ask Mike to help me to do it. I was really uncertain whether he would say yes, knowing that he's got plenty of other things going on. And I was like blown away, delighted when he did say yes.
Very cool. That's a neat story. Anything you wanted to add to that, Mike?
Well, I do remember meeting Vaughn. My only uncertainty was whether we had met more than one time before that. I think we had.
Yeah.
Okay, so I'd love to talk also a little bit about your individual areas of research and how that has fueled your passion for this project in particular.
Well, I have a pretty diverse past. I started out actually as a limnologist. That's the study of lakes, not appendages. That got me thinking very broadly because limnology is a lot of chemistry, physics, geology, and biology. And that was my first job in academia, but I quickly started apparently getting interested in genetics and evolution. I was attracted to genetics because of the more exact nature of the models as opposed to community ecology modeling, which I was starting to wonder how general the results were. So I got into that for about 12 years or so, and then people started sequencing genomes, and we finally had a glimpse of what was actually in a genome. I mean, this is not that long ago, but back in the early 80s, people were taking bets on how many genes were going to be in the genome, and there were bets as high as a million genes and so on. So we were quite surprised. And this started to make clear all the diversity of genomes and the complexity of eukaryotic genomes compared to prokaryotes. And so that got me thinking about all these ideas about non-adaptive changes at the genome level in us big multicellular things. And I guess I started running out of things to do, so I was trying to figure out what to do next, and it seemed like the whole area of cell biology was the big gap in evolutionary biology. We just sort of jumped over the cell for some reason. And so now I think this is a really fruitful area to, not just for me, but for many people to be working on over the next few years.
So it sounds like a general interest in how things came to be, what they are today, driving both your background and your interest in this project and how you were involved here.
Yeah, not only how things came to be, but also what future evolutionary paths there might be given the constraints that we have in today's cells.
Yeah. Very interesting. And how about you, Vaughn?
So I had the just good fortune to see changes in bacterial populations when they were growing on surfaces when I was a postdoc. And it looked like these cells were not forming these biofilms that may be working differently together than by themselves as clones, like I had been studying as a graduate student and before. And so sort of carried that forward as I started my independent career, and it got me thinking a lot more carefully about multicellularity, life on surfaces, how that worked, how is that maybe a model of life's origins, how does it work today, to what extent are bacteria actually used typically multicellular versus solitary, probably more the latter than the former. So even though those are not evolutionary units, the transitions between, being alone and together are quite fluid. So Julia Schwartzman in today's talk did a really nice job of talking about that continuum, and that came out in one of our colloquia report, a part of our reports, really well. But for me, then I had the good fortune to work with a NASA astrobiology project led with a team of folks that Mike knows well, and that just started to motivate us to develop model systems about the origins of multicellularity and basically ask questions about how life might evolve elsewhere, including here. That sort of got me enough into this area that again, this opportunity to take a hard look with the help of experts that Mike and the steering committee put together was like completely compelling. And then more practically, these sort of biofilm associated traits are broadly important in all kinds of applications, be they medical disease, infectious disease, or environmental engineering, or wastewater, or what have you.
Yeah. So that really leads me well into our next question, which is, why is it important to study the history of life on our planet? Why is it important to know about early microbial life?
Well, we're not going to know every detail. We may never understand exactly where life originated on Earth, or maybe not even if it originated here. But the basic principles that are emerging are going to teach us a lot about just basic biology that applies to all organisms. So that's one way I look at it. I think trying to understand the early starting points are also potentially quite valuable in the field of synthetic biology. So right now, synthetic biology it's an interesting area, but it's all based on biology as it exists today. And there's a lot of odd things in today's cells. You probably wouldn't design certain aspects of cells if you were starting from scratch the way they are today. So I think it really potentially opens our eyes up into, exploring it'd be synthetic biology with a big S, not necessarily based on current model system, but trying to design things from scratch. And I think just even developing general theory will help us know what paths to go down, what will work and what will not work.
If I could pick that up, I mean, one of the things that really struck me from the colloquia and then in talking about what we were doing with others was the disconnect between how we think about the origins of life. There's sort of this idea that it's this unitary event, and then everything traced from that. And so we do believe, we have all the models point to a last universal common ancestor, but what this program and others like it have pointed out is that was, and maybe the lucky one, that there were many, many alternative possibilities along the way for all of the major transitions. So, I love to say that to point out what Tom Williams said today is that last universal common ancestor was a darn complicated microbe, actually, that it had already figured out in evolutionary terms an awful lot. And that meant that it had been part of a pretty complicated community of diverse organisms, and you can't help but wonder what those organisms were. And so I think this program really is going to help us peer more deeply into the possibilities of those unfortunate neighbors, but also potentially synthesize them down the road, right? To actually explore what they were like, and were they just unlucky, or were they actually potentially had evolved in a handicapped way that prevented them to be a part of the next major transition. I think if I've learned anything from Mike, it's probably more luck than adaptation, because I think Mike has led us in recognizing the sort of the dominance of chance and relatively non-adaptive processes in explaining where we are. And again, this sort of prospective synthesis can help us appreciate that there's just a lot of chance in the origin of life, and that's both beautiful and kind of cumbersome.
Yeah, absolutely. It is, I think, stimulating intellectually to just think about all of those things that we don't know and what can happen with chance. You've mentioned that we may never know some of the answers to these questions, but something that occurs to me as a scientist is, what are the tools that we have in our toolbox to study something that there's so much mystery shrouded here, and we're looking at organisms that no longer exist. So what are the tools that we use both analytically and computationally to study this line of work?
Well, maybe one example would be, there's this hypothesis called the RNA world hypothesis. Some people are absolutely certain that there was an RNA world, but I think it's open for debate, but that's okay. But that whole thought process really has gotten people thinking a lot more about RNA than people thought about before, and it's developing the therapeutic thing. So that weaves back to your previous question about what studying early aspects of the origin of life, where it might lead. We have general models in evolutionary biology that would apply all the way back to the RNA world. As long as there's a genome, these models apply. So I think we have the capacity to, if the experimentalists tell us they think this is what the setting might have been, we can use models to figure out whether it's a plausible hypothesis. It's not that different than trying to find a geological setting that seems like a plausible place for the origin of life. We ought to be able to at least say it's a plausible hypothesis for how life could have evolved and gotten off the ground. So I think we're pretty well equipped to do that. It's just that there haven't been that many people in our field that have been. Most people in population genetics are focused on sort of modern day issues and conservation genetics and so on, for rightly so, rather than some of these deeper issues. But I think we're really pretty well equipped to do that kind of thing.
You mentioned earlier that you needed a we for this project, and besides the two of you, there was a large group of people in cross-disciplinary expertise that was brought together for this project. Can you explain why this was so important and unprecedented in some ways to bring that full scope of scientific knowledge into this project?
The major reason is that we know that the Earth was really different during these major transitions. And there's been really significant advances in the last decade or so in models of the of Earth, of the chemistry, of the geological conditions, and the changes in the atmosphere, and how that may have influenced the probability of different timing or even different processes of life's origin. So you need experts who can think about that and place the emergent biological processes in the context of the environment of the Earth. That is not what we do. We also need people who are really experts in reconstructing life's histories from genealogical models, phylogenetics. We had key people at each one of our colloquia. We also need paleobiologists who know where to look in the fossil record, know how to explore deposits for possible microbial fossils or major transitions. Synthetic biologists, contemporary virologists. So a reason why we need virologists is that the last universal common ancestor had phage defense systems, right? I mean, so it means that basically, and as Mike said in his remarks, and others have, the origin that those early origins of life, those forms had parasites. So understanding that this sort of Red Queen hypothesis of defense, counter defense, that goes all the way back, and it has shaped all of these processes. I'm sure I'm leaving others out. I mean, there's a lot more to be done in terms of theory. There, we basically relied on Mike, and in terms of a lot of the sort of the virology and models of genealogical evolution in a biological sense, we relied heavily on Gene Koonin, among others. In fact, I joke that we could have just invited, Gene and Mike and maybe Tim Lyons, and we've gotten a good ways there because of how far they are. But I mean, we also importantly had people who are building models today, who are really the future of the field, doing experiments, trying to reconstruct the functions of early life, trying to make it a reality. Very early synthetic biology that's certainly going to mature.
Very cool. So to give listeners an idea, the project included geobiologists, paleogeologists, oceanography, biophysics, bioinformatics, mathematics, cell bio, microbiologists, of course, virology, as Vaughn said, evolutionary biology, and evolutionary genetics, just to name a few. So it was really comprehensive, and it's really, really unique, I think, to see the depth of scientific expertise there. And that was one of the things that really excited me when I was reading through this report, just understanding how that knowledge, it was from top down, bottom up, you're looking at this on all levels and all angles. What is something that you learned from working in this group and in this project that you might not have considered if you were addressing some of these thoughts on your own?
Well, I guess as it became clear what people are thinking about origin of life hypotheses, it did start to dawn on me that, we can actually start developing evolutionary theory for the origin of life. And so I and a couple of my colleagues are starting to do that now. And I guess it's fair to say I hadn't quite appreciated that before. And it's already starting to open up some new questions and new ideas about plausibility that I also hadn't appreciated before until we really started. There's just a lot of arm waving in this field, and rightfully so at some level, but you have to know where to draw the boundaries between, letting your arms wave too widely. And so I think it's been useful in sort of ground truthing some of the ideas that have been thrown around. I mean, we're a lot farther than we were 20 or 30 years ago in origin of life. It's light years beyond, no pun intended, beyond where we were. So it's really, the time is ripe, I think, for bringing this group of people together. And they were really a good group of people that interacted very well and had open minds. So
I'd add that I was hopeful that we would come away with ideas about experimental systems that could be developed. And I realized that there's actually many, many more than I thought that are either in hand or within reach. I do believe, and it does not necessarily require major advances in synbio, even though we know those are coming. You can do that here now by potentially studying communities in extreme environments on Earth that can be made cultivable and replicable. There's several groups that were part of our symposium or colleagues of them that are doing that, and I think that's fabulous. And I honestly hope that funders like the Moore Foundation and many others will recognize the value in supporting the development of these model systems to test these hypotheses.
Yeah, that's really exciting. Both of those are very, very exciting things on the horizon, I think. So I'd like to talk a little bit about our understanding of the origin of cellular life and the evolution of prokaryotic lineages. So if we break it down to brass tacks, what are the basic elements that life forms would need to be viable? I know this is something that you all covered in the report.
Well, it's fairly elementary. You need some kind of metabolism. You need a genome. And you need individuality. By that, I mean there has to be continuity between, a genome and the products it produces and the offspring that come out of that genome. Because if there's no continuity, there can't be any progressive evolution. So one of the early things that had to have happened was the origin of the cell membrane, or some kind of physical setting that maintained individuality. Membranes are also critical to avoiding parasites. Otherwise, you're just putting out the public goods and anybody can take advantage of it. So those are the three main ingredients. And the big mystery is, how did all three of these main ingredients get off the ground at the same time? Did metabolism come first? Did genomes come first? And what about individuality? This is why the RNA world hypothesis has become quite interesting, because now you've got a molecule, RNA, that serves as a genome but can also fold up and potentially do interesting things. So it's also the phenotype. So those are the key things that came early, but it's still uncertain whether the ancestral prokaryote had a completely established DNA genome at that point. So there's still a lot of mysteries.
So some of our listeners may know what the RNA world hypothesis is, but others may not. Would you mind explaining that?
Yeah, so the RNA world hypothesis came out of the conundrum we have. Okay, all of biology today relies on proteins. Proteins carry out most of the catalytic functions in cells, but RNA does a lot, and this why it made the heart of the ribosome, for example, is RNA. There's a lot of other RNAs inside cells that do things catalytically, but proteins couldn't have been fully fledged from the very outset, along with RNA. And then did DNA come much later? Well, we need DNA because DNA is our today's genome, and that's where the genetic code is for all the genes. So the RNA world hypothesis became attractive because it provided a single molecular type that potentially could carry out catalysis, i.e. metabolism. It's made out of 4 nucleotides, so it would also have information content. So it would solve the genome problem at the same time as metabolism. You still need to get some kind of individuality, whether it's lipid membranes or just some porous structures or clay molecules, whatever you have. But that's why the RNA world hypothesis has been so attractive to people.
So what would be some of the challenges of getting life off the ground? You've mentioned a couple of these, but even from a population genetics perspective.
One of the biggest problems is errors. So, and this is one of the problems with the RNA world hypothesis. So there were no enzymes in the beginning, no protein-based enzymes. That's how we copy DNA today. They weren't around yet. And so you would have had to have what's called a non-enzymatic mechanism replication. People have been able to do this in the lab, not really well yet, but lots of different ways to at least get things off the ground. And the error rates are always on the order of zero point 0.1. So that means every 10 nucleotides you're trying to copy in complementary to the parent molecule, 10% are wrong. So this is a real problem, because how are you going to expand your genome to have multiple functions if you can't even produce a 10, 20 nucleotide molecule, pass it on to your progeny intact? So there also had to be in early stages, somehow errors became less of a problem. So possibly an early part of the genome was just a mechanism for minimizing error rates inside cells, and then you'd be able to grow your genome. You still got a problem, though, because your genome is just expanding it's replicating in a linear way. So larger genomes actually have what we would call lower fitness. It's going to take a longer time for a larger genome to produce kids than a shorter genome. So you've got all these internal conflicts that I think we haven't thought about. In today's cells, genome replication is fast compared to everything else that has to be done in the cell. But these were unique problems at the dawn of life.
Yeah, interesting. So, Vaughn, you mentioned that we think that there may have been viruses that helped to shape early microbial life. What do we know about that? And can you talk a little bit more about why we think that was part of this early condition?
Sure. Yeah. So, I mean, the reconstruct, you can model from looking back in time with these sort of phylogenetic models across the genome of what Luca, last universal common ancestor, may have had, and it quite clearly has elements of phage defense built into it, which means that there was something to defend against. And then going even further back, there are some compelling arguments that, the origin of the ribosome may itself have been a parasite that had been co-opted. That we don't know that that's true. But as Mike has pointed out today in his talk, the ribosome is awfully complicated, like way more complicated than it sort of really needs to be, given that the heart of the engine is actually RNA, and there's all this other elaboration around it, which is now baked in, presumably by selfish or maybe even parasitic ways. I mean, there are other reasons why we believe that viruses go all the way back, because, as Mike just said, the best way to gain fitness of a relative to a long replicating genome is to make a shorter one. And we see that all the time. In fact, my student today gave a talk about how this happens all the time in viral infections of bacteria today, where you get competition among viruses within the bacteria that favor these cheaters. The viruses that cheat other viruses. So that's clearly was happening. That went all the way back. And that's an inevitable process.
Yeah, it's so fascinating. Really, really cool. So let's switch gears a little bit and kind of move on to the next evolutionary event and talk about eukaryogenesis. There's many open questions surrounding eukaryogenesis. What are some of those?
Part of the problem is when we reconstruct the ancestral eukaryotic cell, we do that based on what everybody's got in the phylogenetic tree of eukaryotes. Almost everything was there in the eukaryotic cells, the thing we call LECA, the last eukaryotic common ancestor. So the biggest questions then are, what were the order of events? Well, if you don't have any branches below the common ancestor, it's hard to figure out what the order of events would. But one of the biggest questions is about the mitochondrion, the thing we call the powerhouse of the cell. That's how we make our energy. And one of the big debates is whether the mitochondrion had to come early, because without the mitochondrion, there's no way we would have the energetic capacity to build all the other organelles inside cells. Another view is that the mitochondrion came later, and that, say, internal vesicles and other aspects of the eukaryotic cell were there. And so that's still a real unresolved issue. My personal view is that the mitochondrion isn't that kind of energetic savior that some people would like to think it is. But on the other hand, we're stuck with the mitochondria. That's how we make our energy today. So there's no way going back. Possibly the mitochondria originally was a parasite that got somehow tamed by its host cell. And the big thing you have to worry about, I mean, this is a key event, but the thing you have to think about, shouldn't be thinking about this just from the standpoint of the host cell. Because why would a parasite or even a symbiote go inside if it wasn't getting more out of the host cell than it was getting off on its own? So there's a real need to think about the whole question from both points of view, and we tend to focus on the host cell only.
I'd add just another point relevant for our ASM audience is just that the early eukaryote, the last universal eukaryotic ancestor, is a bit roughly equal parts bacterial and archaeal, and the fractions are in some bit of debate. But I mean, I think it's just worth pointing out. I mean, this is known, but we owe all of our replication machinery, the DNA replication, to archaea, but we have bacterial-like membranes, and then obviously we have properties endowed by the mitochondria, which was a bacteria. So we are hybrids. I like to think of it's sort of an inclusive entity that represents all of microbial life, and I think it's then appropriate for these studies to be sort of welcomed back and motivated by the community of microbiologists.
Yeah, it's beautiful in a way when you frame it that way. And that was actually my next question. I was going to ask about the importance of Asgard, archaea. So anything you want to add to that? What is the importance of Asgard?
I mean, aside from the fact that they've got an awesome name and their discovery really revolutionized our understanding of where eukaryotes actually belong in the phylogenetic tree. That's key. And so there's been quite a bit of expansion in understanding the diversity of Asgard-like archaea, and we can now actually get even more precise about which branch LICA belongs to within the Asgard-like archaea. But further, the models of how extant Asgard's work and how they can be cultivated are really based on syntrophies, where to culture them, you have to essentially allow them to collaborate with other microbes in nature. That's how we can see them now, and it's probably likely that that's how they've always been, or many of them have always been. So it then becomes a short conceptual walk about how you get really sustained collaborations between multiple microbes to give rise to eukaryotic life. It's also quite likely that many versions of that happened, right? And again, we are just traced from sort of the one lucky lineage that emerged from that one Asgard-like taxa.
So a key theme that was highlighted in this report is the idea that ecological context drives evolution of multicellularity. What are some of the ecological drivers through which evolution of multicellularity can be selected?
Well, I guess we had a slide of about a dozen different mechanisms. Avoiding predation would be one, just being bigger and avoiding predation on that basis. There are going to be some cases in which partitioning of functions among different cell types. So cyanobacteria they're bacteria. They're multicellular. They have at least 3 cell types. One fixes nitrogen in many cases, but that cell type has to be kept, and it's set up so physiologically it's anaerobic, because nitrogen fixation is an anaerobic process. You don't want the rest of your cells to be anaerobic. So partitioning of metabolic functions among cells would be another one. There might be contexts in which you want to sink more rapidly or less rapidly. If you're in the open waters of the ocean, there's any number of different ecological mechanisms, and then you just follow descent with modification.
Yeah, I mean, certainly dealing with stress in found, well, still on contemporary Earth, but certainly early Earth, being on surfaces with, say salt ponds that evaporate and then are flooded and so on. Multicellularity allows for just durability, and as it does today for many of the taxa that we might use for models.
Makes sense. So after participating in this project and these discussions, what is your sense about whether or not we are poised for another major evolutionary event, and how likely that might be?
Do you mean in terms of actual biology or the evolution of the science? Because I think the evolution of the science is already happening.
Yeah.
As a function of many variables. But so which one?
Let's go with the second. I was thinking the first, but I like where you're headed with this.
Yeah.
Evolution of the science.
So, I mean, okay, I think in terms of the biology, boy, I don't know. Maybe you do, Mike. I think, well, let me talk about the science for a second. We haven't even begun to we're still early, as Julia said, I think really nicely in our panel today, Schwartzman said, we're early in the collector's curve of models of extant multicellularity, for instance, of the biodiversity in sort of the, say, Asgard-like archaea and the role of centrifuges and consortia on Earth today that can serve as models, for instance, of eukaryogenesis. So, I mean, it's possible that we'll actually discover some really, not possible, it's likely we're going to discover some biology that's going to just, blow the tops off our heads, right? But what we know is definitely happening is the evolution of the way we do science. And I do believe that programs like this that bring together cross-disciplinary groups create the opportunity to do kinds of science that many of us weren't thinking about before. So, for instance, you can do experiments that are really well tuned to the reality of the Earth that was available at the process that you're trying to study. Intuitive to some, was not to me. I think that synthetic biology, again, is going to allow us to build some of those what-if scenarios and actually test if they worked the way we think they were, among others. Mike, what do you think?
Well, I mean, evolution is an unstoppable process, right? It's always ongoing. There's no way to stop evolution, but it's a slow process. So most of the things we're talking about, the origin of life, origin of eukaryotes, these are processes that unfolded over millions, if not tens or hundreds of millions of years in some cases. So I don't want to let you down too much, but I think it's unlikely we're going to see a revolutionary change on the planet. On the other hand, we're changing the planet pretty quickly right now, and we are opening up new opportunities that haven't been around for a while. So we will be seeing some dramatic changes. The places you'll see it mostly are in pathogens, I think, with very rapid life cycles. And of course, we've seen that in the past few years. Those aren't major morphological revolutions or anything, but they are, from the standpoint of the virus, they're sort of what we would call a peak shift almost. Yeah.
Well, when we're covering this many years of science, there's so much we could talk about, so much more that we could discuss today. But instead, I will turn listeners to our report. Again, if you visit asm.org/MTM in the show notes, you can access the report directly. That will be it for today's show. Mike and Vaughn, thank you so much for speaking with me today and for taking the time to discuss this early microbial life project.
Thank you so much.